The examples I was saying, because I know you were saying immutable destiny, you're talking about the law of monophyly. So obviously, like the Lenski experiment, those generate, because obviously we know that bacteria have very short generation time, so we could actually see millions of years of evolutionary history based on these experiments, right? But all I'm saying is I find it funny that the big evidence would be bacteria producing, more degenerated bacteria. Like, just kind of, I'm going to send him the email. Like, just give me your input on that. I'm just kind of curious.
Well, I'm not sure particularly what you mean by degenerated. It's the fact that they're able to develop proteins to survive within, very extreme conditions for them in relatively short time periods is what we expect of evolution.
Well, the thing is, it's, these E. coli have shown because, even under the most favorable condition, right? Because it's lab conditions. So we would say that those are a little more favorable, say, than out in the wild. But if we're looking at, say, mutation and selection, which is obviously the only game in town for evolution, it's not really, bringing forth any forward as in, germs to genius evolution, because even the big thing, the citrate digestion that they've adapted from, it was, due to the already existing information, but that gene, it was permanently turned on and it damaged the genetic switch. So it was an advantage, but it was a degenerated advantage. So I'm just saying that after all those generations,
Well, I mean, you can't take a holistic approach to it. Evolution is only talking about like like we were saying earlier about the whole, in animals in an Arctic as opposed to if it's in the Sahara Desert. The
yeah.
bacteria
So like,
is in isolation with its environment. So if you're saying that something benefits with its environment, say in the Arctic Circle, then that's a benefit. And of course, if we put it in the Sahara with the advantage it would have in the Arctic Circle, it's not going to fare too well. So you can't expect it to be universally beneficial.
but I'm just saying, like, if we're, and once again, I understand, the law of monophyly. I'm just saying, if we're seeing, even after this many generations within the lab, if we're just seeing, bacteria produce bacteria, why should we believe in bacteria to biologist evolution? I mean, if we don't see that type of gain in the information systems, is it scientific to believe in, say, universal common ancestry? Go ahead.
Well, I mean, you do know that some bacteria don't always stay as well, don't always grow independently, right? That there's some bacteria that actually favor and that will almost always stay as, in essence, a colony of bacteria. They will operate as more or less as a pack.
Yeah, just like you can find, a colony of human beings, working together as a pact, but it's still a bunch of human beings working together. It's still a bunch of bacteria working together. So what is the point of that?
Right. But so the thing is that when you have plasmogamy and karyogamy occurring, so when you have the fusion and sharing of cytoplasm and DNA between cells that are particularly functional towards a specific task, say it's metabolism.
Are you talking about horizontal? Are you talking about horizontal gene transfer?
That might be it. Been a
Well,
while
like
since
they're,
I looked into it, but
yeah, because they're like, bacteria is transferring some of their genetic material or information to other bacteria within that colony, correct? Is that kind of what you're
yeah,
referring to? But the thing is, if you got information specific to a bacteria transferring, through horizontal gene transfer to more bacteria, you're still only going to get more bacteria. So I mean, that's no help to your
well,
bacteria to
no, but I mean,
biologist.
you missed the first point I made, and that is if these bacteria specifically function towards a specific goal, say metabolism, right?
Yep.
In one part of, whatever large colony they're part of, then you'd expect it to be the case that whatever genes are responsible for the metabolism that are responsible for producing, cytochrome or anything else that's responsible for metabolism is going to be selected for, and that these genes are more than likely going to be transcribed very frequently because of the fact that that's what they're doing. So you would expect to see change within either the structure or how the evolvability of ATP, things like that. So yeah,
So,
you would see.
okay, so are you seeing, new novel information being added to the genome, or are we just seeing, the alteration or the tweaking of, say, the existing, genetic information? Because obviously, like I said to Athena earlier, tweaking the genome of, say, a bacterium, whether it's through duplication or mutations, and then whatever type of mutation it is, it's not really going to result in a human genome, right? Because you're still acting on that pre-existing information. Go ahead.
Well, do you agree that for existing information to be added on, that there has to be information to begin with?
Yeah, so you would have to have, well, because in our model, right? Because if you're looking at models, we would say that, God put significant pre-existing heterozygosity encoded into each created kind. So if we see changes based on that pre-existing heterozygosity, that's consistent with our model. But the thing is, we would predict degraded changes, and that seems to be what we observe are degraded changes. Because would you say that, beneficial mutations, if they exist, are they rare?
Sure, they're rare, but that really doesn't do anything. All it means is that you're dealing with a numbers game.
Okay, so let's assume that, these near neutral mutations, let's just say they're accumulating in our genome much like rust accumulates on a car. If they're neutral and natural selection can neither act on it or against it, how is natural selection going to get rid of these mutations that are building up not only in our personal genome, but in the population? Go ahead.
So, I mean, number one, the one issue you're having is that even if all there are certain amounts of genes that are present within a genome, but that doesn't mean all of them are going to be transcribed necessarily, right? And then not all of them are going to be transcribed at the same rate. That's why epigenetics exists, because of the fact that we know that certain patterns.
Actually, just one sec, because I posted the link, because for some reason that guy that wants to come in, the email wasn't working, so I posted it into the chat. So if he makes his way in, I can then delete the link. Did you make your way? He is in? You want to say something, brother? Okay.
Oh, I appreciate all the work you're doing.
Hey, man, how you doing? Here, I'll delete. Good, brother. Thanks for joining us in this brief hangout. Are you a creationist as well? Like, are you anti-evolution?
Yes, but I'm not a young Earth creationist, though. I'm more of an old Earth creationist. But okay. I am against evolution, though. Okay, so you're more in, say, are you in the Hugh Ross camp, right? Because I know Hugh Ross, he denies, and Frank Turek, they deny large scale evolution. Okay, that's good. You could say that. I'm more in the gap theory too. I believe that there was, like a stasis, extinction, and then a real fast, rapid, or speciation after that.
What makes you?
What about the evolution?
Okay, so back to evolution, like Immutable Destiny. Like if we look at, so I guess we could kind of talk about that all day. But if we actually look at, say, specific, because would you agree that mutations are the engine of evolution?
Well, not specifically. Mutations in the environment are usually.
I understand, but like obviously natural selection plus mutations plus selective pressures, environmental pressures. But I mean, ultimately, it is mutations that are adding the new information, right?
Well, once again, that's not really. It's an answer that requires both of them, because something will act upon a mutation will be selected for under the assumption that the gene is expressing and that a mutation can occur within that gene, that the gene's not already being silenced. So yeah, I mean, if the environment is favorable for that gene to be expressed within an organism, then sure. But if you're in an environment where some particular gene's actually being selected against, where the organism doesn't need it to be expressed, then you're not really going to expect to see a change too much.
Okay. As I was explaining to Athena earlier, because calculations would dictate that, molecules to man, large-scale evolution would need, roughly 8 beneficial mutations, say, per year for 3 billion years. But do you think we observe that type of mutational increase, I guess, within that, even per year? Because that's the numbers needed for evolution to achieve within a human history, say, to reach the 3 billion DNA letters we now have. So that actually seems that 6 billion would be even worse for evolution. Like, are you familiar with Haldane's dilemma? Like, do you find that to be a problem for forward evolution?
I believe I forget what it actually states. I can look it up though. But most of the DNA that we have is regulatory DNA. At least we believe that to be the case.
Well, like if we look at, like for example, if we look at that junk DNA, right, that you were, because now we're seeing that, in that non-coding DNA, it contains sequences that act as regulatory elements, right? So that means they determine when and where genes are, say, turned on and off. And also those elements provide sites for obviously specialized proteins. I think those are called transcription factors. So they'll bind, and then from there, they'll either activate or repress the process by, say, which the information from genes is turned into proteins. So would you agree that those are pretty important functions?
I would say they have function, but technically speaking, they're not functional.
Okay, so what I just said to you, you don't believe that that's functional, because in those non-protein coding regions, we see those regulatory functions. That's not function to you?
So the reason I want to consider it functional is that what's functional in an organism is the proteins and the various structures that are made as a result of synthesis of proteins. If you don't have something being synthesized, right? I mean, sure, you have regulatory elements, that's fine. But these regulatory elements don't do anything on their own. They're acting as a way to guide polymerases and transcriptases to monitor the activity of these genes, which that's perfectly fine. But it still doesn't transcribe a gene. It doesn't.
Now, do you think you're okay? Now, do you think you're putting a little bit of an evolutionary bias into that assessment? Because if these regions of non-coding DNA do provide instructions, say for the formation, which we just agreed, for the formation of certain kinds of RNA molecules, like even CRISPR and I were talking about how non-coding DNA includes, and he admitted that this was actually functional, it includes transfer RNAs, right? And even ribosomal RNAs. And what does that do? That helps assemble protein building blocks, which is amino acids.
Sure. Yeah, I don't think anybody denies that RNAs are significant. I mean, the thing is that one gene can create several hundred different products, and that's mainly because of RNAs.
Okay, so obviously it's not idle then. So these non-protein coding regions are doing something. They're not idle. So in our model, that's consistent. And based on the fact that we know very little about the DNA code and the functions, because we're accidentally coming across various functions. So imagine we actually, say, did these gene knockout tests. We're only going to, obviously the trajectory is strong, say for genome-wide function. And I think based on your assessment, I think that's more of an evolutionary bias, because to me, and I think to the majority of people, I mean, these are important functions, and any type of mutation on a gene that has a regulatory function will be deleterious. Go ahead.
Like I said earlier, I mean, I'm not denying the fact that it's not important, but if you're talking about function particularly, that doesn't matter, because you're talking specifically whether or not a portion of DNA is coding for some kind of product, which ultimately is a discussion that takes its roots in biochemistry. The really simple aspect of it is that, look, sure, there are things that are, that allow for polymerases to activate and to create a product. That's, sure, it acts as a regulatory sequence. It helps guide RNAs into place. But in the same sense, though, it's also not coding for anything. So to some extent, it's functional, but not strictly within a biochemical setting.
I think Well, for one, I would just disagree and say that, everything that I've been saying and we're agreeing on, I would say that is functional. I would say that a mutation on a gene like that would, create a problem for the organism, as it would be like a ripple effect, right? But I think it all comes down to models, right? So for evolutionists, the ultimate, and correct me if I'm wrong, the ultimate cause of genetic change is mutation, right? Like if you look at these nuclear DNA differences and these mitochondrial DNA differences, you would conclude those are based on mutations, correct?
Well, part of it's mutation, but that's not the most important aspect of it.
Okay, now for us though, and I'm sure you understand, like what would like, for example, pre-existing heterozygosity, like I understand you don't agree with it, but what do we mean by that when we say that?
That there are genes that exist on, different chromosomes that we replicate via, that we can't replicate on our own, that we require the use of another human being to replicate.
Okay, yeah. So like millions of differences, say in the DNA sequences of chromosome pairs. We're saying that was built into organisms from the beginning by the creator. Okay, so when we look at the origin of species, we're saying that long periods of time is not required for the new species, because if we start with these heterozygous organisms, the visible distinctiveness and change in offspring, it's going to be fast. So if you look at things like recombination, gene conversion, that would provide many new varieties of, say, combinations of traits quickly, because we're saying these differences are already built in. So if we go back to functional versus non-functional, non-protein coding regions, we predict that, they're not going to be idle. They're going to have function, because if we're saying that these tens of millions of nuclear DNA differences were built in, if they're design variation, we'd expect them to have action. Okay, so obviously ENCODE revealed that 80% is transcripted, and now we're seeing more function. Like for example, pseudogenes, which we thought were false genes, actually show function, or something like protein moonlighting. We're seeing these proteins that have multiple function. That's consistent with us, because that's a prediction, a testable, falsifiable prediction based on pre-existing heterozygosity. But you're saying, obviously, these are not true functions, or it's spurious transcription. To me, that's a rescue device. How do you feel about that?
So it comes down to how you view something as functional. I mean, if you're considering the fact that one gene is transcribed in the course of, 8,000 runs as being functional, sure, that isn't really a valuable definition of functional, because it means that at best that, okay, there's a gene transcribed, but it's far from being transcribed regularly. There's some genes that are transcribed, 30, 40 times within an hour, let alone 30, 40 times within however long the duration of the experiment is.
So are you familiar with Mendel's experiment on the pea plants?
Yeah. The one thing Mendel proved is what he got wrong.
Well, he proved, because he was looking at the principles of inheritance, right? So he found concerning, and correct me if I'm wrong, but he found concerning traits that they actually remained distinct, as in they didn't blend, but they appeared and disappeared in family trees, right? They came in dominant and recessive types. So what I'm saying is...
You are familiar with the history of Mendel, though, right?
Well, I know him and Darwin. He didn't know genetics, and neither did Darwin, but he's seen this based on the experiment he did.
Are you aware that Darwin also did experiments in genetics?
No, but he didn't know of he didn't study genetics. Everything, obviously, genetics came about. The field of genetics came about after both Mendel and Darwin.
Sure. I mean, Mendel is considered one of its founders. However, even with Mendel, part of it, the fact is that when other people studied Mendel's work, they noticed that it was almost instantly incorrect, because there's information that was left out.
Okay, I just want to clear something up. You agree with me that Mendel and Darwin were ignorant of genetics, though, right?
Sure. I mean, genetics was just becoming to become a thing. It was in its infancy.
Okay, so he did see, though, that these traits they appeared and disappeared in family trees. They came in dominant or recessive places. But now that we look at a genetic level,
sure,
we know why that's to be. Is what I'm saying, that this is consistent and it is indicative of the pre-existing heterozygosity that we believe in regards to the origin of species. Because, like I said, heterozygotic DNA sites will provide that necessary variation. We look to Mendel, to provide the necessary evidence for that. Obviously, that's all I'm saying with.
Well,
with Mendel.
There's a lot of things wrong with the experiments he did. The first of which is the fact that, one, he ignored evidence, and one of the evidences he ignored is the fact that a very specific amount that would be predicted by what we consider modern, Mendelian genetics, is that when he breeded certain plants, there were hybrids between the original and the parent, which is the same thing Darwin observed. And also, what was failed to be noticed by Mendel, and couldn't have been known at that time actually, was the fact that he was particularly examining a particular thing in genetics.
Okay, I just want to stop you right there. Are you saying that his pea plant conclusion based on these traits, recessive and dominance, that's not universally accepted? Like, if we look at the pea plant conclusion, are you saying that's not universally accepted?
The result
because we're focusing on the pea plants here.
So when he published his results, they were not initially accepted.
I understand that because they didn't know genetics back then.
Well, people had been doing these experiments for a little bit before then, and they understood kind of the rough aspects of it. But people didn't accept it for a few reasons, and one of those reasons was that he concealed data, and the other reason is that when he was dealing specifically with a phenomenon in genetics known as independent assortment.
Okay, but are you saying that what he observed in regard to family trees, with the traits, are you saying that that's not, like, I just wanted to, do you agree with that part?
So the mechanism he observed within pea plants is highly non-characteristic of many organisms that we do see.
Okay. So what, and give me a comment on this, what Mendel proved and is today accepted, I'm saying this is accepted universally, is that a new trait can emerge in just one generation based on his pea plant and pre-existing differences. So we're saying if the created kinds and Adam and Eve had these pre-existing differences and pre-existing heterozygosity, change in new traits can come about quickly. Go ahead.
Yes, it can. However, the crux of that is, once again, when he was performing his experiments, one of the interesting things about it, and there's been molecular studies on this experiment as well, because they've recreated it, is the fact that the genes of interest for Mendel were significantly spaced out, which what that means, unfortunately, is even if an organism inherits, there's a very fair chance that an organism will inherit one gene and still maintain the other, its original genes. In most organisms that we observe, however, that's not the case. Most genes tend to overlap with one another. So if I inherit a mutant form of one gene, I'm more than likely to, how do I say this? I'm more likely to take on or to inherit other genes that are mutant.
Well, it just sounds like to me that you can't grasp pre-existing differences, because obviously in the evolutionary literature, mutations are the source of all differences, right? Because
yes, it does. Okay. So the big difference, though, is that if we're dealing with, like we were talking about before, with coding and non-coding, right? Mm-hmm. Genes are regions by definition, where proteins are coded for and have a function. If there's significant difference, like if there's significant spacing between these regions where genes can be expressed, well, of course, with crossing over, you would expect that some genes get passed on, some genes don't. You're going to have some kind of distribution that determines that.
Yeah, but I'm saying with the pre-existing, because would you agree that human chromosomes differ by, say, millions of single nucleotide polymorphisms, like SNPs, between and within individuals? Would you agree with that?
Yeah, I would think so.
Okay, so if we look at DNA sequences, say in a chromosome pair, for example, we know they're, let's say they're identical. Okay, they would be said to be homozygous, right? So, but when they differ, even by one base pair, then they're said to be heterozygous, right? So I'm saying inheritance and not mutations is the main cause of heterozygosity within individuals. And regardless of all the details of Mendel, he did demonstrate recessive and dominant traits. So that's all I'm saying. And I think that that is a basis and a starting point for what we're saying in regards to the origin of species.
So here would be my question then. Would you say that there are certain combinations of genes that like, you know, I can have different forms of a gene, but they can all be considered homozygous?
Okay, so you're saying you're going to have multiple variants of the same gene, but at the same time it's homozygous?
Right. So what I'm saying is that it may be possible for, you know, a particular coding region of DNA to be selected for because there happens to be a certain, you know, a certain pattern of nucleotides that favors the transcription of that particular gene into this phenotype, as opposed to another gene on another chromosome being expressed due to something like codon specificity or.
Okay, yeah, I understand. Okay. So what you're saying is all genes have heterozygotic gene sites and homozygotic gene sites. Like there's no actual population that's just purely homozygous or purely heterozygous, right? Is that what you're saying?
Yeah, effectively. And
I would say that that's you know pretty basic. But if we go back to our model with pre-existing heterozygosity, say encoded into each created kind and into Adam and Eve, that speciation coming from there, obviously, you know, we're going to see mutations afterwards, and we're going to see, you know, degradation and sorting out of information, and it's going to go from more heterozygotic DNA sites towards homozygotic DNA sites. But of course, there's still going to be both, right? All genes have both.
Well,
I agree with that.
The only way you would get homozygotic gene sites is that if humans somehow decided to or started to learn how to lay eggs. I mean, because of the fact that human beings, when we reproduce, our DNA combines with the DNA of another person.
Correct.
So the chance of mutation in that a beneficial, you know, some kind of beneficial mutation will occur is increased because of that, as opposed to in a homozygotic situation, you know, if I'm laying an egg and the DNA I have is just replicating and creating copies of itself. So, okay. I mean, as long as people keep, you know, reproducing in a heterozygotic fashion,
sure.
I mean, we've talked earlier that, sure, you're probably going to have a lot of bad mutations, but you're also going to have a few good mutations that are going to get selected for and passed on through the generation.
But we can get a heterozygous gene site if the animal didn't migrate and get isolated. Because what we're saying is the pre-existing differences, you know, they were far more heterozygous sites than homo at the beginning, right? Because would you agree that a small heterozygous population will tend towards homozygosity? Like when this happens, and this kind of goes back to the Mendel pea plant, you know, example and experiment, some recessive traits will be revealed. And it sounds like the creation and evolution models both agree that all species within families share a common ancestors. And we also agree that if you don't, let me know.
Well, so I mean, here's the thing though, right? Is that like with human beings, there are more than one species of human before we wipe the majority of them out. But things like plants, for instance, things like other mammals, there are other species and there are other classification of phyla that species can breed within.
Okay, well, obviously I don't.
Like if you're restricting it to breeding within only its own species, you may have a point.
Well, would you agree? Okay, would you agree that the parental generation would be more hetero than its offspring generation?
Not necessarily.
Okay, the majority proves the rule. Would you say most of the time?
No.
Okay, but you just agreed that a small heterozygous population will tend towards homozygosity, but now you're saying that it's not the majority of the time? Like, I don't understand.
So generally speaking, right, in the behavior of animals, usually incest isn't usually incestuous relationships, usually incestuous behavior isn't favored among reproductive successiveness. So usually you will have organisms that, wander a great distance, decide to lay eggs, another species comes along, or another, family of fish that are completely unrelated come along, fertilize the egg, and you have an increase in heterozygosity.
So if you look at, say, our model of, and I'm going to comment on that too, but I just want to just make it clear to everyone listening. Like, if we look at our model of pre-existing heterozygosity, would you say that science, you need to have testable and falsifiable predictions, right?
Sure.
Because, okay.
That could be done fairly easily, right?
I just want to let you, because obviously, we've talked in detail about, the non-protein coding regions, pre-existing heterozygosity, but we're saying that obviously this most of the non-coding DNA will be functional, okay? And obviously we would probably agree to disagree that what we're seeing is functional. I would say that it's clearly not idle, but here's the big thing, right? Differences in, say, mitochondrial and nuclear DNA sequences between families are expected to reflect functional differences, but not just due to neutral mutation. Okay, because if we believe in original created kinds, we're going to agree in function. Okay, genome-wide function, but different functions across families. Because we would say that the created kind is, you could probably take it up to to the family level, but measured mitochondrial DNA mutation rates and known mitochondrial DNA differences, say within families, they would be expected to fit the creation timescale and what we would assume is a constant mutation rate, because the created kinds, and obviously if you understand the model, they're obviously going to start with no mitochondrial DNA mutations, right? Because we inherit mitochondrial DNA through what? Our mother, right?
Okay, I think even if you're going by, creationist standard, you would still assume that I wouldn't grant you that there's a constant mutation rate, because, and again, we seem to both agree to the fact that the environment can cause or can dictate the rate at which mutation can be affected.
But would you say it's fair that if evolutionists are allowed to inherit mutations, say as the source of all variety, why can't we as creationists inherit pre-existing differences? Like, would you say that that's fair if we're comparing models?
Well, I think you can, but I think that generates more harm than good for you. Because if you do genomic studies across species, you'll find that a lot of species, humans and apes, and there's quite a few, like mammals, for instance. We share quite a few different. We share quite a lot of genes, and some of them we don't even use. Some of them we really don't have a function for, but they're there because we call it evolution. You guys would call it, I don't know.
Okay,
but the point being is that we share genes that aren't even within our own family sometimes.
So are you saying that the environment determining factor is natural selection? Would you be able to make a prediction based on natural selection? Because if we look at these constant mutation rates in regards to mitochondrial DNA, the differences would actually point more towards a 6,000 year time scale versus a 200,000 year time scale, say back to the origin of humans. Go ahead.
Right. So if I'm going to argue that, like, temperature, for instance, or the environment can play a crucial role within the mutation rates, I can subject mice to all sorts of different temperatures. I can have some colony of mice that starts off, give them a very happy life, food and water and all the things that a mice needs, And, after a certain amount of time, I segregate them and I say, okay, so one of these mice are going to live in normal conditions that mice normally would operate under, but some of these mice are going to operate under conditions that would resemble more of a drought situation. So they get less water at certain increments. There's more heat that they're exposed to. They have significantly less rest time. I can create parameters to test, what are factors that could increase mutations within these mice.
Okay, so can you fill in the blanks, say, of how much natural selection then has to eliminate in order to, say, fit within the evolutionary theory? Like, would you be able to sufficiently fill in those blanks and then make that successful prediction?
I'm not sure if eliminate by meaning like silencing of certain genes, or if you mean just out and out eliminate, the gene itself.
Well, I'm just saying that it seems like there's a lot of guesses, which means no testable prediction, which would indicate that it's obviously not scientific. Because from my understanding, most nuclear DNA differences, if we're looking at, say, equids, between them, they're homozygous within those species. So if we look at the 26 million nuclear DNA differences, let's say between the imperial zebra and the domestic horse, just 2 million are heterozygous within the domestic horse. And we actually see that other species display this same pattern. So if we actually look at all the nuclear DNA differences, they're significantly more than obviously the mitochondrial DNA differences. And we would say that's because of pre-existing nuclear DNA differences. And we would say that the mitochondrial DNA differences are based on a constant rate since Eve, right? Because we pass it down from Eve. Go ahead.
Well, yeah, I guess to some extent that would hold. I mean, the first part of what you said I don't agree with, because of the fact that, sure, it's hard, but that's also why people are invested in the field of science. I mean,
well, how much? Okay, well then I guess it's simple then. How much mutations does natural selection have to eliminate then? Like, obviously, for it to be testable and falsifiable, and you need to fill in the blanks, and you need to make a prediction on natural selection, how much mutations does natural selection ultimately have to eliminate? Go ahead.
I mean, you're asking, okay, so in a controlled experiment, we may be able to guess that.
Are you currently testing it, or are you saying that this is the prediction?
So what I'm saying is that, like, if I were to set up an experiment with mice under conditions that I knew, I could probably make reasonable predictions about how many mutations occur per generation or something to that effect. But for me to say, here's how many mutations needed to occur for, this mice to go to something that's, a different species or something like that. I would need sufficiently more data about the environment, about, how these mice were living, what kind of pressures are they under, things like that. I wouldn't have enough information to judge that.
Okay, so what you just said is you can't, and it's only a guess. Now, let's look at the family of obviously cattle, sheep, antelope, because this is one that we can actually look at, okay? Because they have been analyzed. So there are hundreds of species in this family for which there is ample nuclear DNA and mitochondrial DNA information for analysis, okay? Now, if we look at it, and I urge you to do so, there are only a few thousand mitochondrial differences separating any two species within this family, which is amazing, because what we do then is we draw a branching tree from these data, okay? Now, what the tree shows, it shows splitting, okay? So the distances between the splitting events, let's just say the branch points to new species, are reflective, obviously, of the number of mitochondrial DNA differences. Now, what I said earlier, we're assuming a constant mitochondrial DNA mutation rate, okay? Now, the plot shows the number of species as a function of time. So what the plot suggests, if you're following me, is that there is a constant mitochondrial DNA mutation rate within families, and the mitochondrial DNA mutation rate per species decreases as homozygosity increases. But for nuclear DNA, and I'm almost done, speciation decreases heterozygosity and hence the potential for speciation. So if we actually look at the species that have been analyzed, it's more consistent with with our model of pre-existing DNA differences and constant mitochondrial DNA rates. Go ahead.
Well, sure. I mean, that's all well and good. The first question, of course, is, you're dealing with you're dealing with some amount of species. That's fine. Certain species of animals have different means of survival mechanisms. Some are engaged by sex arrangement. Some are engaged by hyperactivation of certain chromosomes. Of course, the thing that has to be asked is this constant just throughout this particular selection of species that were chosen, or is this constant throughout all species?
Well, obviously, I mean, we've only sequenced certain species. I'm looking at species.
Well, sure. I mean, so, I understand that. But of course, though, that's fine for the sample size that they have, but it's still speculative.
And that's our prediction, right? Because my whole point is mutation rates of a handful of species. The whole point of everything I just said is they are consistent. That's how creationists, but not evolutionists, can make predictions on all animals' mutation rates. So whether it's the pre-existing nuclear DNA differences that we say are based on pre-existing heterozygosity or constant mitochondrial mutation rates, we're making the predictions. And so far, the trajectory is pointing towards a stronger model based on a biblical-based model. And I understand that we'll see what the future has in store, but some of these testable predictions are coming true, which I'm actually posting in the description box. But it seems like the trajectory is stronger for us than it is for evolution itself. Go ahead.
And I think a few evolutionists have suggested the same thing, that mitochondrial DNA differences may be the contributing factor towards speciation. The question, of course, is because it's young, how much of a bearing does it have on in the rate of speciation? I mean, it could be significant, but it could also contribute 1%, which in that case, that's fine, but that means there's something still larger at play.
But if the DNA differences, say, of all species so far, obviously we haven't sequenced them all, if they're too few based on everything I'm saying, evolutionists have always predicted far too many differences. So why is that the case? And after you explain to me why that's the case, would the rescue device be natural selection? And if it is natural selection, I want a testable, falsifiable prediction as to how many mutations natural selection has to eliminate. And if you can't give me a testable, falsifiable prediction, then it's not scientific. Go ahead. Because it sounds like we're agreeing that based on these mitochondrial DNA differences and nuclear DNA differences, the creation model is the one making the testable, falsifiable predictions, and the trajectory of the non-coding protein regions is pointing towards genome-wide functionality. Now, obviously, we'll see what the future has in store, but what I'm saying is, based on all this current evidence, the creation model appears to be more scientific. Go ahead.
Yeah, I mean, again, it depends on how much data gets concorded. It may be the case that certain species of animals have higher than average, or very nonlinear trends for how their mitochondrial DNA mutates, and things of that nature. I mean, it's a matter of speculation.
And what I like about what we're talking about here is this is what Dr. Jensen and Dr. Mays were supposed to talk about. They're supposed to have a very scientific discussion on testable, falsifiable predictions that were found in replacing Darwin. And that's what we're doing here. And this is how we advance science. So I think for us to do it this way, in a giving each other critical feedback, looking at the empirical evidence, looking at the data, looking at these rates within mutations, whether it's mitochondrial or nuclear DNA, I think this is the best way to say discuss the models, right? Because to be scientific, it has to be testable and falsifiable. And if we're making testable and falsifiable predictions, and we're actually seeing so far that it's consistent with the empirical evidence, I guess it's a matter of perception, but it's my perception and opinion that the creation model better fits the data. Go ahead.
Well, I mean, it seems that you accept the majority of evolution. You don't like calling it that.
Well, I would say obviously that with pre-existing heterozygosity, limits, genetic entropy, a sorting out of information, we're going to expect more, obviously, if we're looking at traits, functions, features, you know, we're going to say that those are more based on a degeneration and a sorting out of information. And I think that that's pretty evident that that's the type of changes we see. But I mean, if you think otherwise, you know, obviously that's okay. You'd have to document obviously thousands of information adding mutations to get your bacteria to biologists. And I don't think that that's, you know,
I don't know necessarily if you'd even need thousands. You could require merely hundreds, because once again, I mean, the particular way that genes are expressed and how products are formed are the process of alternative splicing, as we found out within, I believe it was probably 80s, 90s. Well, the other thing is that what seems to be weird is that giving pre-existing heterozygosity, as you like to claim, that we still have and have far more bacteria in an organism than we do of our own cells. Like your own body, for instance, has about 10 times the amount of bacterial cells from other organisms than you do of your own.
And how would you say that that would be inconsistent with, you know, everything that I've said in regards to, you know, the successful predictions, the successful mitotic, and I'll answer that question because I'm just trying to say that, you know, we as creationists, we're now using evolutionary assumptions on these mutation rates, right? Because the constant rates would be, and I want to know if you agree with that, that would be an evolutionary assumption, right? If those rates are constant. So in regards to everything I said, and then in regards to what you just said about the bacteria, why would that be such a hurdle or an inconsistency to that data? Go ahead.
So what seems to be a hindrance is the fact that, like, in at least through the course of evolutionary history, which I know you don't accept, but what appears to be the case is that bacteria in archaic organisms were the name of the game back, you know, when the Earth was still burning and was allowing life to form. But what's curious is, according to the endosymbiotic hypothesis, and it's all but shown now, is that organisms throughout their history seem to depend upon, you know, so even though heterozygosity is a means for organisms to survive and thrive, we still rely heavily on homozygotic organisms to take care of synthesizing certain proteins, to create certain genes, to do certain things that our own bodies really can't handle.
Well, how does that relate, though, to pre-existing differences? Because now, judging by what you're saying, it just seems like you're still not understanding the pre-existing differences that we're talking about, because there's nothing wrong with homozygotic organisms and homozygotic populations. I'm just using that as a way to show you how our model predicts the origin of species ultimately. And then when we look at these mitochondrial DNA, obviously mutation rates versus the nuclear DNA differences and those rates, that matches up perfectly with what we would expect, predict, and retrodict. Go ahead.
I mean, I think it's like anybody who's at least examined like history of Earth would come to the conclusion, even from a biblical model, that it seems to be the case that if God created anything first, it was the bacteria.
Well, obviously, you know, because even myself, I take probiotics, right? Because bacteria is fundamentally good, and even E. coli is fundamentally good, but it's that it's still based on that degradation and mutations within even, say, because obviously we can both agree that, you know, there are plenty of good bacteria. The majority of even the bacteria that you would think and look upon as negative, like E. coli, is majorly good, right? It's good for our gut. It's good for our gut flora. It's healthy for us. So in a biblical model, that bacteria was necessary. That was a necessary creation and design by God. But based on, say, genetic entropy, based on the fall, degeneration, a lot of this bacteria has descended into a state of negativity, obviously, within the bad bacteria, especially, the big reasons why you get sick if it's a bacterial infection. But originally, it wouldn't have been like that. And that would have been the same with viruses, because we know some viruses are actually helpful as well, which is funny. And this is all easily explained by the fall and by, obviously, degeneration, of course. Go ahead.
Well, sure. I mean, I think there's other things that just become interesting as well. Like you have organisms that only breed or selectively breed through the use of like planting eggs into another organism. Like fleas and flies are notorious for doing this.
No, and
you plant like thousands of eggs into a host organism, wait for it to hatch, and all but destroys the organism that's hosting it for it.
Yeah, no, and these are, obviously, we're living in a fallen world. We take that back to say, a point of least entropy, a point of least degeneration. I mean, I think it's pretty consistent within a biblical history. But and obviously, I don't mind you responding to that, but I want to kind of engage, because I know we got Warning in here. We got a couple of people in here. We just had a really, I think, good discussion there, but I want to see what their input is. Like, hey, Warning, how you doing, brother?